Hello, and thank you for tuning in for this virtual seminar with the Engineering Biology Research Consortium. You can learn more about the EBRC by visiting our website, ebrc.org. Today's talk is by Kate Elston. Kate Elston is a graduate student studying in Jeffrey Barrick's lab at the University of Texas at Austin. She's currently pursuing her PhD in microbiology and is broadly interested in the use of synthetic biology to alter relationships between insects and their symbiotic bacteria. Her talk is titled Engineering Aphid, Symbionts for Food Security. Thank you so much, Kate, and I'll turn the time to you.
Alrighty, thank you, Becky, and thank you for the introduction and for the opportunity to speak. I'm glad I've still got a chance to do so, even though we've all been separated into our homes. So yeah, as Becky said, I am a graduate student at the University of Texas, and I work with Dr. Jeffrey Barrick. And today I'm going to be talking about some work that I've done engineering an aphid symbiont for food security. All right. So just to give a brief bit of an introduction, I'm sure many people are familiar with symbioses that humans have with our gut microbiomes and all the bacteria that live on our skin and in our body. And we're probably pretty familiar with the fact that these are digesting components of our diets. They're working to protect us from pathogens. And sometimes things can go wrong. We can have commensal bacteria that then can overrun and become pathogenic. And so a lot of work has definitely been done in that human microbiome. But as we probably know, there are a lot of microbiomes out there in the world, and insects are no exception to that. So insect symbioses take many forms, and then they can kind of be neatly described by this little figure that I have right over here on three sorts of axes. So the z-axis over here is the age of the relationship itself, so how long a bacteria has been co-evolving with its insect host, and that will affect some of these other two axes. So over here on the y, these bacteria can range from being essential, which we call obligate, to less essential but still providing benefits, and those ones are typically facultative insects, and those are less essential for survival than the obligate bacteria are. And then on the x-axis here, these relationships can range from parasitic to mutualistic, so either harming the insect or providing them with some sort of benefit. And so I'm just going to walk you through a few examples of each of these types of relationships. So the first one that people may have heard of before is this bacteria Wolbachia, which Mike did mention in his talk kind of vaguely. This is the bacteria that people have been using to try to control mosquito populations because it causes sterility in these insects. But if you're more familiar with these bacteria, yes, there's a caveat that Wolbachia isn't always parasitic. This is just a great example of one that people may have heard of, at least, that falls into this category. So then bacteria that maybe have been more evolved with an insect and have developed it to have a mutualistic benefit, we tend to call those facultative symbionts. So these are ones that are not essential for insect survival, but they do provide some sort of benefit. So the example that I have here is a bacteria that is a symbiont of weevils. And what it does is it gives that weevil that's colonized, it gives it a tanned and hardened cuticle, which is beneficial for that insect to actually survive in harsh environments, but it's not essential for survival. The insect can live without it, it just might not be as fit. So those are facultative symbionts. And then for bacteria that are more essential for survival, have co-evolved with their host for really long times, those are obligate symbionts. And hemipteran insects like aphids have really characterized well-characterized obligate symbioses and relationships with these bacteria that they have co-evolved with for millions of years that actually live inside of specialized cells in their body. They provide them with essential nutrients, and not only are they essential for the host survival, they are also reliant on the host for their own survival. So that's just an overview of some common relationships between insects and their bacteria. But kind of the next question, of course, is, okay, there are these relationships. can we do anything with these relationships to kind of help ourselves out? Can we manipulate them in some way? And yes, people have done this. So a lot of Mike's work was focused on transgenesis as a technique to alter insects. So that's directly engineering the insect itself. But my talk is focused more on paratransgenesis, which is engineering the symbionts in order to change aspects of the insect. And so one of the first cases where this was performed was back in 1997 by De Rossela's group. And they engineered bacterial symbiont from the kissing bug to express antimicrobial peptides that killed off the parasite that these bugs carry that causes Chagas disease. And a lot of examples of paratransgenesis kind of fall into a similar category. They're trying to control some sort of vector for human disease or some sort of agricultural pest. And that's been done over the years, kind of sporadically. It depends on the insect and how easy it is to manipulate the symbiont that actually lives inside of it. Recently, there's been a bit more like buzzier examples. There was this one where they engineered a bacteria that lives inside of a mosquito gut, this Serratia AS1, to express an antiplasmodium effector protein. So that's to control the spread of malaria. And then if we think about other things that we can do with these sorts of bacteria, you can also think about ways that we can engineer them to protect insect health for beneficial insects. So we had some great work out of our lab by Sean Leonard that came out just recently, where he engineered a gut bacterium of the honeybee to express double-strand RNA that target viral pathogens and parasitic mites that usually would harm these bees. So yeah, that's just an overview of what's been done. You can think of a whole bunch of different reasons that you might want to engineer a symbiont to manipulate an insect that maybe is not as genetically tractable through transgenesis. So just to dive into my insect, I work with aphids, which people commonly know as major agricultural pests, but they are valuable to study for both biologists and the agricultural industry. So the reason why they are major agricultural pests is that they carry a lot of plant viruses. You can see here, this is a sorghum plant that has undergone an aphid infestation. You can kind of see some discoloration there on the leaves, and the head of this sorghum plant hasn't been able to flower like it normally would. And aphids kind of act like little mosquitoes of the plant world. They feed directly on the plant phloem, and they're able to spread viruses from plant to plant really easily. And as you can see up here, they reproduce very quickly, and they can quickly overtake a plant as well, which also is going to cause problems. But beyond that, they also have a variety of interesting biological traits. So they have plastic traits. So I mentioned that they reproduce quickly. That's because for most of the year they are asexual, but as photoperiods change, they'll produce offspring that can reproduce sexually. And then those offspring will lay eggs that then can only reproduce asexually again. So kind of cool biology there. They also have winged and non-winged morphs. When they get really stressed out and crowded like this, they'll produce offspring that have wings that are able to fly and go feast on a new plant. And they're also great models for symbiosis, which is great when you're trying to engineer them for the purposes of paratransgenesis. So I'm going to dive into that those symbiotic relationships real quickly here. So as I kind of mentioned at the beginning, there are obligate symbionts and facultative symbionts. Aphids have this obligate symbiont that provides essential nutrients to it and is housed in those specialized cells that I mentioned at the beginning. But they also have facultative symbionts, which can be one of three that I have listed here: Serratia symbiotica, Hamiltonella defensa, and Regiella insecticola. And these bacteria are also vertically transmitted from parent to offspring, and they're found inside of those same specialized cells, the bacteriocytes, but also throughout more of the body, like sheath cells and hemolymph, or the insect blood. And they provide secondary benefits to the host, like tolerance to heat stress or protection from wasp parasitoids. And these facultative symbionts are the ones that were actually able to colonize a lot more easily than the obligate ones, which are very reliant on their host for survival. So one of those, Serratia symbiotica CWBI 2.3T, which is a very catchy name that I'll have to be referring to it by, it was recently cultured from the black bean aphid by a different group that we were able to generously get this strain from. And that was cultured from the black bean aphid. All All right, so then how would we go about actually developing this as a tool for paratransgenesis? And that's kind of the goal for all of this work. And those are the things I'm going to be walking you through today. So first step, of course, is we have a bacteria. It has not been engineered, and we need to learn the compatibility of tools and techniques with this particular strain and know what we can and can't do. And then once we've engineered the strain, need to establish a method for actually delivering this back to the aphids, characterizing the dynamics with which it colonizes the insect itself and how it's transmitted from generation to generation, and then exploring the versatility of the tool in more than one aphid species, because ideally you'd like to be able to use this in a variety of different species. And then third, of course, we want to actually test an application for this tool. And in the case of mine, as you may have gathered from the title, that application is kind of a pest control potential use case, which I'll go into more detail later. Okay, so first things first. Yes, the strain can be engineered pretty straightforwardly, and actually it's compatible with a whole bunch of different commonly used techniques. So I've been able to transform it through both conjugation and electroporation. It's compatible with a whole bunch of different origins that range from low to high copy number, as well as constitutive and inducible promoters. And we've also been able to integrate into the genome itself and generate a GF pre-expressing strain, which is really great for actually tracking the aphids that we then feed this strain, because you have that glowing effect going on. So yes, very easy to engineer this strain, which has been fantastic. And now the next step, of course, is to feed it back to aphids. So this is the general protocol that I typically follow when feeding it to aphids. I will first grow up my engineered bacterial strain with that GFP integrated into the chromosome. You'll see me abbreviate this as SSGFP throughout most of my talk. I spike that into an artificial diet, which contains a bunch of sugars that the aphids will then feed on. And then I move them back to the fava beans, which they are a lot happier and healthier eating on than the diet. And after about 3 days, you can basically take those aphids, put them on a blue light plate, and you'll see them light up with that bacteria, which is fantastic. We can feed this bacteria to the aphids, and we can easily see which ones are actually colonized with our engineered strain. So I've done a bit of optimization to this technique over time. So initially I was doing these experiments with blue food dye spiked into their diet, so that I could mark which aphids were actually feeding on the diet. And from those experiments, I was getting colonization rates at about 26%, which is not ideal for actually doing further experiments. That's pretty low. But kind of more recently, I've optimized that by actually using yellow food dye instead of the blue food dye. You can't tell which one's ate, but it actually serves as an attractant, and we get much higher feeding rates. So about like 24% of the aphids that feed are colonized, which is a lot better, and you can do some great experiments with that then. Okay, so then kind of digging into characterizing how this bacteria actually colonizes the aphid. This is where we kind of notice some differences from that canonical facultative strain that I mentioned earlier. So you may remember that I said that that strain colonizes those specialized cells of the insect. We have not seen that at all, and that's been corroborated by another group who doesn't have the engineered version of this strain. But what we can see really clearly with our engineered one is that our strain colonizes the entirety of the aphid gut over time. So this is just a time course of images starting on day 2 after colonization up to day 10. And you can see that over time, this bacteria basically spreads throughout the entirety of the gut. This is a view from the top of the aphid, and this is a view of the stomach side of the aphid. And you get these really lovely images out of this. But what we know of this is that the strain is colonizing the gut, so anything that we want to express from the strain is going to be released into that gut environment. And this has some advantages and disadvantages over at colonizing those specialized cells. But then, as I mentioned, of course, we want to test this in a few different aphids. So the aphid that I've been showing you pictures of is the P aphid, which is a model aphid species that is most commonly worked with. And that's the one that I've done most of my work in, but we've also tested the colonization in this cow pea aphid, Aphis crassivora, as well as the black bean aphid, Aphis fabi, that was originally isolated from. And you see this same pattern of gut colonization. And if we actually quantify this, you notice that there is a lot of consistency in the pattern of this colonization, where it increases over time in all of these different species. On the y-axis here, I have the log 10 of the average concentration in each aphid, with CFU per aphid being the units there. And it tends to reach a titer of about 10 to the 7th to 10 to the 8th CFU per aphid, and that's pretty consistent for each species. But you might notice that there is something weird going on day 10, and that kind of ties into my next point that, you know, if we're engineering this strain, we need to know how it's actually affecting the aphid itself before we throw in any other toxins or anything. And what we've seen is that although Apisum, that model aphid species, the P aphid, survives really well up to day 10, lives a normal lifestyle, Aphis pavy has some impacts on its mortality, and then Aphis crassivora, we actually definitely are reducing the longevity by colonizing it with this strain. This is that cowpea aphid. And over time, once you hit about 5 days, most of those aphids have already died after just being colonized with our GFP expressing strain. So not an ideal aphid to probably work with on any further studies, but it is important to take into account how this strain just baseline is affecting the fitness of these aphids. So luckily, the P aphid, that model aphid, it does not show the same pattern, and it will colonize it stably over its lifetime, no impact on longevity. So we're able to go forward with that. Okay, and just one final thing that we wanted to test was, and where we've seen kind of a discrepancy from the canonical strain, is in the transmission of the strain. So if we're gonna colonize aphids with a strain and think about applications in pest control, we need to know how it's transmitting from generation to generation. And what we've seen is that it's not faithfully transmitted to offspring. So very few aphids will receive the symbiont from their parents. Over here, I have a chart showing the number of offspring colonized, which is in this darkened region, out of the total offspring that each of the parents produced. From both the F1 generation, which was fed on that diet spiked with the bacteria, and the F1 generation, which comes from this population of colonized offspring of that F0 generation. And in both cases, we don't see really high transmission rates. And we have hypothesized that basically it is spreading through the environment itself. So when I look at these aphids, I'm not seeing colonization initially right as they're born. Sometimes it appears in later stages. We also see our glowing strain of bacteria appearing in the aphid waste product, or honeydew as it's called. So their waste glows bright green, and this might be a potential method of transmission. And as with some of the patterns that we see in the gut, this has been supported by some work by other groups. So our strain is not what we initially thought, but the fact that we've been able to engineer it has helped us to characterize the dynamics more and think about how we can actually apply this when we perform our applications. Okay, so getting into the application, as I mentioned at the beginning, I kind of had a pest control sort of focus for this application. So basically I've engineered this strain to contain a plasmid that it has an inducible toxin on it, so that when I feed it to the aphids, induced expression of that toxin, we should see an increase in aphid mortality. So the toxin that I chose is this UCP1 protein from Pantoia stewartii, which is a bacteria that colonizes plants that aphids can feed on, and it is pathogenic to them. And the way that it does that is it forms these little bacterial aggregates in the gut, and that will clog up the aphid's gut and cause them to die. And there was some work by another group who characterized this that indicated the expression of this protein by E. coli could cause a similar effect, although there wasn't a lot of data on that, so we were curious to see whether we could apply it to our setup. So the way the protocol works, pretty similar to the one that I showed you before. I grow up the bacteria to log phase, feed aphids on diet spiked with that bacteria, move them back to plants, and then after 3 days, after that bacteria has gotten a little bit of time to establish, I'll feed them on the inducer, which in this case is arabinose. Okay, so then to test whether this arabinose induction system would work, I tested both in vitro and in vivo. And in vitro, we do see strong repression when there is no arabinose, and then good expression of GFP in this case when we are inducing the aphids with arabinose with that protocol. Or sorry, this is just the bacteria. And then when I feed it to aphids and I feed them on diet without arabinose and diet with arabinose, you kind of see that similar pattern as before, where the aphids who have this bacteria and then fed on that inducer glow, and if they haven't fed on the inducer, they don't glow at all. So you get great repression and then expression when you're actually inducing it. Oh. All right, so what I did is I took the construct that I know works, swap in my toxin protein of choice, which is that UCP1, and basically repeated that experiment with a similar setup, only this time expressing a toxin as opposed to GFP. And we were excited to find out that you do actually get a significant increase in aphid mortality when you have this plasmid and you induce expression of the UCP1 toxin. So all the aphids that I'm showing in this chart, for simplicity's sake, have been fed on that arabinose inducer. And in red, we have the line that shows the survival curve for the UCP1 aphids, and then in gray you have the ones that do not have that toxin plasmid. And we've been able to achieve significance of greater than 0.05%. You may notice that both of these aphids are dying, which is not ideal, and that's kind of a byproduct of having to feed them on artificial diets spiked with this inducer. But I have repeated this experiment multiple times, and consistently every single time I get a significant increase in the mortality for the UCP1 aphids. So that's very exciting that we're able to engineer a bacteria that naturally lives inside of these aphids, get it to express a toxic protein, feed it to the aphids, and actually see a significant effect. So just kind of thinking about the future then with this, I've already kind of outlined this potential path here, which is engineering a native bacteria to produce a toxin for use in pest control. We've also got some other work from another graduate student in our lab, Julie Perot, who's using this to learn more about the aphid's relationship with this bacteria. I already kind of talked a little bit about how the bacteria that we found is a little bit different than the canonical strain. She's doing some work to try to express proteins that might be involved in symbiosis. And then kind of more long-term lofty goals, I've been striving to try to have this bacteria engineered to produce double-strand RNA like Sean Leonard did in bees in order to do RNAi in these aphids to reduce vectoral capacity for viruses that are harmful to plants, and also to improve aphid functional genomics. So hopefully I didn't run over too much on time. I would like to thank all the members of the Barrick Lab. Special thanks to Jerry Maida, who is actually not in our lab. He's another graduate student at UT now. Julie Perot, who does a lot of other work with aphids. Peng, who has helped with a lot of synthetic biology stuff and developing these constructs. Sarah, who's our lab manager, who helps me with a ton of stuff. And Sean Leonard, who developed a lot of the work that helped me to do this in aphids as well. And then all of the Moran lab, who I collaborate with a lot because they are experts on insects. So thank you very much. Taking questions.
That was great, Kate. Thank you so much. And thanks to all of you for tuning in. We'll call it a day with that, but anyone can learn more about the Engineering Biology Research Consortium at our website, ebrc.org. And make sure to check back to our YouTube channel for more great talks and other great educational tools.